my institute, so it's not totally unfamiliar site. Also, Estonia is pretty far from Brazil and a lot colder than your place. Still, probably very good place to study mosquito-borne viruses, because half of the year we have 0 mosquitoes. Other part of the year we have plenty of them, and they are large and mean and plentiful. Okay. Now I need to find out how to make this actually work. Okay, I actually managed. I start from the main thing, people who actually did the work. So this is a few years back of my research team, and I have collaborators from everywhere. If I couldn't list them, they are on this image in this corner, which I think also shows Brazil, so probably not Sao Paulo region. So let's start from my favorite groups, which are, of course, positive strand viruses. And to be short, they are just numerous and very diverse. They exist in different groups, depending who and how systematize them. They can find quite a lot of variety. I'm mostly interested in this alpha virus supergroup, but we also, of course, pay attention to flavivirus supergroup. And like pretty much everybody these years, we can't, of course, escape coronaviruses. So, starting from alpha viruses. Alpha viruses is the only genus inside a family Togaviridae, and generally are assumed to consist of viruses which infect vertebrates and are transmitted between vertebrates by mosquito vectors. This is called the classical alpha viruses, which we do know most about. But that is, of course, not all, and they are actually quite diverse. From vertebrates, they can infect birds, reptiles, mammals, and the hot blood animal infecting alpha viruses can be broadly kind of divided between old world alpha viruses and new world alpha viruses. Also, this is more kind of biological than geographical division, because yes, a lot of these viruses indeed occur in old world, but also there are Mayara virus, which definitely originates from South America, and Burma forest virus, which is from Australia. New world alpha viruses are usually associated with encephalitis in their mammalian host, and they are Western, Venezuelan, and Eastern equine encephalitis viruses. There is also less known that there are alpha viruses which infect aquatic species, and most prominently fish, and the main player in here is salmonid alpha virus. Also, it has several names. And increasing number of alpha viruses have no known vertebrate host. Probably they don't even have them, and can be regarded as insect-specific viruses. So Eilat virus was first discovered, but there is Typhorist virus, there are aqua salad virus, and many others. So it's a broad picture. And last but not least, there is also a rock band called Alpha Virus, which doesn't make a very good show, but has a very nice titles of their songs, like Trauma Model, Prosthetic Limbs, Kill Anything That Move, and so on. So we are focusing on the real viruses in our talk. So alpha virus infection cycle is typical of that of positive strand RNA virus, which starts from binding to receptor. There has been huge boost of understanding or increase of understanding what the receptors are in last 10 years. After the binding to the receptor, virus is internalized, releases a capsid, and capsid releases a RNA. And from RNA, a viral replicase precursor called the polyprotein B123 is produced and then processed to the mature replicase by a very well-organized cascade. Then this binds to a genome, generates a negative strand RNA, which stays in duplex with its template, and is used to generate a lot more genomes and shorter subgenomic RNAs. The process is super active. In a permissive cell, there is somewhere around 200,000 copies of genomic RNA what is made, and maybe closer to 500,000 of subgenomic RNAs. Subgenomic RNAs are used to translate structural proteins, which together as a genome form a nuclear capsid first, and then also mature virion. So there are many aspects, and we have been involved in studies of pretty much everything on them. But our main interest has always been around of this one, how the replication occurs. More specifically, what does this viral RNA and what does this replicate? Well, it is kind of a molecular machine, and we really want to know how it works. So, of course, in reality, it's not something like a bar. This is a very well-organized proteins, and there has been a significant progress in last, say, 20 years in understanding what these proteins are. The protein number 2, which is the largest among the viral non-structural proteins, is a multifunctional one. It was believed to be a protease and antipase, and also then shown that it actually is a both. And indeed, somewhere about 20 years ago, the crystal structure of alpha virus protease was resolved, which resulted in quite a stunning finding that they actually consist from 2 domains. One of them has a classical protease fold, but other resembles a methyltransferase, but it is not active. But it is still crucial for a viral enzyme to function. It took a lot longer to reveal the structure of the N-terminal part, which is a RNA helicase antipase domain, and which has the classical Rec-A domains, but also something very unique on this beginning of the protein, specifically folded N-terminal domain. And it is capable of binding RNA, which we are going to talk a bit more. So NSP3 is a mysterious protein. It consists from 3 domains. 2 of them have a globular structure. The macro domain is owing its name because it's similar to macrohistones. And alpha virus unique domain, also known as a zinc binding domain, is binding zinc ions. And well, originally was not really thought much about. This is the red part in here. But quite recently, a French team, together with Spanish team, resolved its structure already not on the small individual molecule, but the macromolecular aggregated forms. And it is clear it forms disc-like structures, and more specifically also filamental structures. So the scaffold of these structures found in cell is a zinc binding domain. The hypervariable domain has no structure whatsoever, but instead it has a bunch of linear peptides which can bind to host factors. So basically it's a binding hub for host proteins. Finally, NSP1 is another ring, which is really looking nice. It's a dodecameric structure. It's bound to membrane and seems to have a pore inside, which, well, probably is not empty. This was originally thought that's a capping pore, but there is something sitting in. And final player, this took longest to resolve, was viral RNA polymerase, NSP4, which not surprisingly has a classical right-hand fold, like any other RNA polymerase of virus, but seems to be a very highly disorganized. It has a lot of flexibility and looks really ugly on a crystal structure. Very difficult to find out what it does. And there is a reason why it looks so disorganized. It needs a partner. And the partner is actually NSP1 and NSP2, because high-resolution analysis made in infected cells and also the cryo-EM and X-ray crystallography on a reconstituted replicase complex shows that the NSP1 ring, this pore, is not empty. It contains the NSP4, which in this state is perfectly folded. And on top of it, on cytosolic side in here, is sitting NSP2 helicase, while the double-stranded RNA is sitting in here in specific microorganelle called a spherule, which looks like a bag. And on top of it, there is an NSP3 ring. So if we are really looking, that it probably looks like that on top view, that there is NSP3 ring and below is NSP1 ring. And this is a side view how the replicase complex looks like. So it's a fascinating architecture, and we kind of know a lot how it looks like. But what we are struggling to understand and still don't understand in full is how it actually functions. So to study it, it's quite a challenge. We have a lot of theories how these complexes are formed. This is one of them. We know that the precursor of mature replicating protease is involved, but not exactly how. We know that there are a lot of host factors taking a part, but putting it all together is, well, very challenging work. And one of the issues with this is that you can't really use freely a mutagenesis approaches, because whenever you mutate something in a viral genome, it results that when you put your virus into cell, unless it's dead, it can easily revert to the original form, and you are not really getting much information what you have done. To overcome this issue, we decided some 10 years ago that we need some system which cannot revert, which is easy to manipulate, and can be used in whatever lab, not also in a high biosafety level laboratories. So this is alpha-viral genome. If you truncate it down and taking only the critical parts needed for replication, which is this beginning of the genome, middle part of it, and 3' end, then you can substitute rest with some kind of reporters and got them mini genomes. Of course, these can't replicate because they don't produce anything. They don't encode any replicase protein. But if you make the replicase protein from another RNA, either the full 1, 2, 3, 4, or in 2 parts, 1, 2, 3 plus NSP4, you actually can reconstitute a replicase. So how it works? You usually deliver 2 plasmids, or in some cases just 2 RNAs made in test tube to a cell. If they are plasmids, they go to the nucleus. They are transcribed by cellular polymerases. One mRNA is producing a replicase, and another is translationally silent because of lack of cap. But the replicase can bind it, make a double-stranded RNA, and produce a large amount of capped RNAs, which is either full length, thus mimicking a viral genome, or shorter, mimicking a viral subgenomic RNA. And if they incorporate markers, you can monitor replication, the genome synthesis, and transcription, which is a subgenomic RNA synthesis. And that is quite easy. This system is modular and easy to use, and actually applicable also to screening and analyzing antiviral compounds. So easy to use system. And it has been our working horse for, well, about a decade. To tell all which we have found out would probably take a huge amount of time. So I truncated it down to a few main points. Number one, I already mentioned that viral 5' end is really important. We know that it contains a very specific secondary structures which can be resolved by using a method called shape, and then a folding of the structure. So there is a massive number of stem loops And these stem loops are absolutely crucial. Some, like this one in beginning, if you destroy it, the virus is absolutely certainly dead. Other stem loops are less critical. Some are important for mammalian cell replication, some for insect cells. So there is a lot of differences. So it is chikungunya virus. If you look, of course, not every alpha virus resembles chikungunya virus. So Semliki forest virus complex, so this is the relative of chikungunya, have almost identical fold at the 5' end, while a virus is belonging to other serocomplexes have likely a different ones. So this, of course, begs the question, how do viruses recognize the template RNAs? Do they recognize each other's template RNA or only their own? And the answer actually was already pretty much known. Alpha viruses can, to some limit, recognize other RNAs. Say, Semliki forest virus can recognize chikungunya, and so on. But what is the rule? And this we tried out using this system. We took chikungunya virus RNA, we provided it different replicases, and yes, chikungunya can perfectly replicate its own RNA, but myrovirus does it also very well, while Venezuelan equine encephalitis virus and actually all New World alpha viruses are really poorly at this job. If we take RNA of Sindis virus, which is not related to Semliki, you can see that all Semliki forest virus-like viruses are happy to use it. Of course, Sindis virus itself, but also Barmah virus, forest virus. But again, not these guys. And if we take Eastern equine encephalitis virus RNA, you can see it's also pretty universal. The only virus which cannot effectively use it is ALAT virus, which is insect-specific virus. So always they recognized by their own, but if the template RNA comes from a Semliki forest virus complex, it is relatively poorly recognized by other viruses. So there is something. We actually went down and find out what this something is. And the something is that probably it's a specific fault at the 5' end. This is a swapping experiment. I'm not going to the details. It's pretty boring. It's basically, it tells that the very, very beginning of 5' UTR makes a difference why Semliki forest virus group templates are recognized mainly by related viruses and not by more distantly related ones. So this was actually not a major discovery because something like that was already shown by Sindis and Semliki forest virus. But if we ask one step further, which component, what protein recognizes the RNA? It becomes really confusing, and currently we don't have a few answers. So real answers are missing. So we have made experiments to narrow it down. We have provided the template RNA and used the swapped replicase, which consists from 2 parts, 1, 2, 3 part, and then SP4 part. This is very easy scheme where you can actually use a heterologous and homologous combinations. Of course, we generated all possible versions of the templates and checked which replicase, which component can recognize them and use them. And the answer is pretty easy. Whenever the replicase template matches with P123, it works. So the template recognition is in here. So, okay, we are now smarter. We have eliminated 1 out of 4 proteins, but we still don't know which 1 out of 3 is the guy. Also, we have suspicion, supported by 2 lines of evidence, that probably NSP2 is a crucial factor for template recognition. Why do we think so? First, this is the only protein of alpha viruses we know for fact from crystal structure that it definitely interacts with RNA in very specific mode. The RNA binds into the groove of the protein, and surprisingly, it's not electrostatic interactions which are most important, but hydrophobic. stacking interaction between nucleotides, the aromatic parts of nucleotides, and 3 aromatic amino acids in NSP2. So we know that if we disturb these interactions, we have very little impact on antiphase activity. So this is not needed for antiphase activity. But we know that when we mutate any of these critical residues, virus is absolutely dead. We cannot get anything out. So it's lethal for a virus. So maybe it's a template recognition, maybe it's a helicase activity, maybe a combination of them both. We can't tell for sure now. So it's a complex. So at least we have some evidence that NSP2 is the key. Now I switch gear and go to the NSP4, which definitely is not the major player. Also, it doesn't exclude it completely from RNA recognition. This is a catalytic subunit of polymerase and most conserved protein of the virus. So we decided to check whether we can produce NSP4 in cells, generating NSP4 expressing cell line, and then transfecting it with a template RNA and the missing part of replicase 1, 2, 3. And as we expected, if we have match, this is chikungunya NSP4. If you provide chikungunya, onyongonyong, RRV, Semliki forest virus, or Mayara virus, 1, 2, 3 regions, and template, it works. If you provide a new world alpha virus part, it doesn't. We did the same trick with NSP4 of Eve, and it, of course, as expected, preferred its own P123 and template. So it looks kind of play, but you actually allows you to do quite a fancy things. One of the things which you actually can do is, for example, you can delete NSP4 from a virus genome. You generate an absolutely dead virus, no polymerase, and if you transfect it to the normal cells, it does absolutely nothing because it can't. But if you transfect cells which express NSP4 and induce them, you can easily see that it actually produces significant amount of viral proteins. This is a capsid protein in here. And if we did the same design with Eastern equine encephalitis, it works the same way, except that, of course, it preferred the cell lines expressing NSP4 from Eve. So you can actually generate the viruses which lack NSP4 totally and rely on NSP4, which is made from cell. So the same actually worked with NSP1. We tested. I'm not going to go through the details. We couldn't delete NSP1 like we delete NSP4. But we can express it. We can make compensatory changes. We can make a virus which is lethal. It can, virus harbors lethal mutation. It cannot replicate in a normal cell. But in cells where we actually produce NSP1, we get efficient replication. We can rescue a virus. We can infect a next generation of cells under this line. It's looking capsid protein. And the virus is actually happy. This is chikungunya virus. Defective, it can't replicate in cells where there is no NSP1. It can't replicate in cells where NSP1 is from Eve or Syndis. It can replicate very well if it's its own NSP1 of chikungunya, but it's pretty much working also with Rosqueva and to small extent with Semliki forest virus. So I think it's pretty useful tool. And useful tool comes to some kind of antiviral research. You know, everybody have working with antivirals at some point of time. And to my opinion, most of Brazilian virologists try to do it one way or another. And there is always a kind of issue. Well, in Brazil, chikungunya is BSL2. So is Mayara. But Eve is not. And if you are wanting to work with the dangerous viruses, it's much easier if you can use this kind of safe viruses which are restricted to certain cell line. For example, chikungunya virus lacking NSP4 but expressing non-luciferase marker in wild-type cells does pretty much nothing. It enters, it translates a little bit from incoming DNA, RNA, non-luciferase, but that's it. But if you have a production of NSP4, then you will get a normal replication as a virus should do. And if you apply viral inhibitors to here, we tested it with 3, they actually work. Ovataclax, for example, is an inhibitor of virus entry. It blocks endosome acidification, while 4-fluorouridine is inhibitor of NSP4 polymerase activity. So you can get exactly the same result as you get with a wild-type virus. If lacking NSP4 works exactly the same way, in a cells which express this protein, it is a normal virus, and you can make the same antiviral assay. And you can find out sometimes quite surprising. For example, 4-fluorouridine is indeed also powerful inhibitor of EVE, but ovataclax does absolutely nothing to EVE infection, which means that EVE membrane fusion upon entry requires different conditions, and it's not so critically affected by acidification of endosomes as it is in case of chikungunya virus. So now I will go to slightly different topic, but still stuck to alpha viruses. Alpha viruses have 4 replicase coproteins, and even so, they are very, very multifunctional. They definitely cannot do everything what is needed for virus infection. Each and every one of them interacts with host proteins, and NSP3, most of them. It's interesting if you look on literature with what NSP3 interacts. It depends what virus you're using and what paper you're reading. You usually have very little overlap between proteins identified by one method or another. But if you are really working with chikungunya virus or Semliki forest virus, something sticks out. G3PP, one protein which goes on, BIN, CD28P, and FHL1 are something which you usually pick up regardless what assay you are using. So I put 2 different examples. So we can conclude that these proteins are really important. And these proteins are important because their interaction with host or with virus is actually crucial for viral replication and can be used for attenuation of viruses and generate the vaccine candidates. For example, NSP3 is phosphorylated. In case of chikungunya and onyong virus, this picture shows radioactive blood. No matter what you exactly delete or change, the protein is still phosphorylated. So it is scattered. It's not one site of phosphorylation, but many. And if you remove them all, you kill a virus. But if you remove part of them, you can actually see that the phosphorylation deficient NSPs are not interacting with all host factors. Sometimes the interaction with CD2AP is lost. Sometimes the interaction with BIN is weakened. Sometimes some interaction is increased. Sometimes we get somewhere in the middle. And here I picked FHL1 as example to show how important it actually can be. FHL1, is it needed for infection? The answer shortly is no. Virus can actually replicate quite well without it. But if you knock out FHL1 gene in mice and infect it with chikungunya virus, well, if you use a wild-type mice, it develops disease, which is characteristic biphasic swollen of the joints. But if you use FHL1 knockout, then nothing at all. RRV, not affected. Myaro, not infected. Onyong virus, severely affected. So 2 viruses are requiring it. Not exactly for replication, but most likely they require it for pathogenesis. So if you do it other way around and knock out the ability of virus to bind FHL1, which we did, we made a mutant viruses, which wild-type virus binds FHL1, mutants don't. So, and checked these viruses. They all replicate quite nice in cell culture. But if you put it on chikungunya virus, if you use this chikungunya virus lacking FHL binding and infect the mice, no symptoms. But it generates a very nice immunity. So this is another type of attenuation which you actually can use to generate an attenuated viral genome. Not kind of deleting part of viral proteins like in case of this e-chick vaccine virus, but by making just a mutation, preventing interaction with some crucial host factor. So alpha viruses are really cool and interesting to work. But there is also one more aspect which I would like to briefly highlight, which is an alpha virus infection cycle. It requires mosquito. Mosquito transmission is something which is crucial. The overall easiest and most common idea is to kill mosquito and virus is gone. But can you mimic something, use a molecular tool to make mosquito unable to carry on the virus? And here we used a phenomenon called superinfection inclusion, which in many alpha viruses is mediated by NSP2. The diagram actually shows that if you express a viral NSP2, this is a black bar, the viral replication goes down, and NSP2 of Sindis virus, for example, can affect Sindis virus itself and many others. So it's a kind of big panel. So of course, cell culture is cell culture. Whether it works in mosquitoes, can it be used is a big question. And together with our collaborators from UK, we actually demonstrated quite convincingly that yes, it can. This is just a demonstration of superinfection inclusion. If you infect cells, mosquito cells, with a virus expressing the red color, cherry, and then try infect them with a virus which expresses a green color, it really doesn't work. Green fluorescence is very weak. So its cells are kind of refractory. So we mimicked it by making a transgenic mosquitoes, a group in Purbright in UK. And they obtained mosquitoes expressing either NSP2 of Sindis virus or chikungunya virus. And you can see that especially prominently in Sindis virus case, the mosquitoes cannot very well acquire infection by chikungunya. This is a virus which is used. If the protein, what mosquito expresses chikungunya NSP2, they are not as easily infected by Sindis virus, and Sindis virus own NSP2 has a strong effect. If you look it further, you can actually see that the viral titers are much lower if the mosquito expresses a viral protein. And if you try to look how much virus reaches a mosquito saliva, you can also see that it is reduced a lot. So this strategy can be applied to make transgenic mosquitoes which cannot spread the virus. But it is, of course, very tricky, very difficult to make mosquitoes which are actually viable and would survive anywhere outside of lab. Last 10 minutes, I will really focus on two other groups of viruses, flaviviruses and, well, coronaviruses. Flaviviruses, I have no really big experience of studying, except we really wanted to use the same strategy that we used for alpha viruses to generate refractory mosquitoes, because like alpha viruses, flaviviruses also encode a protease. But it didn't work at all when we tried to apply this approach. And hence we generated a really, really crazy system together with the people from UK, which consisted of transgenic mosquitoes, which express, one hand, they express a Cre recombinase, which is bound to the membrane and not released. It is sitting there and doing nothing. And they carry a lethal effector, which in our case is scorpion toxin, which is not expressed because it is lost. So mosquito carries kind of ticking time bomb, but it's never activated. The trick in here is that the key can be released by a protease coming from incoming virus. So it looks really sophisticated circle. But when we actually tested it, to our not small surprise, it worked very powerfully. The mosquitoes in here shown infection with the surrogate, a chest virus expressing a Cree. Every mosquito which carries this kind of ticking bomb dies in a very short time after infection because the Cree produced by virus activates the scorpion toxin. But also Zika, which doesn't produce any Cree, but produces a protease which releases a Cree from a membrane, causes the death of these mosquitoes. And much earlier than death, mosquitoes actually stop flying. They kind of drop to the bottom of the cage and move erratically but can't fly. And the mosquito which cannot fly cannot transmit the virus. So this is a kind of technology which is possibly applicable for many flaviviruses. Finally, I really would have liked to avoid the coronavirus, but we can't. Because for me, coronavirus is intrinsically related to Brazil, because Brazil is the first place whenever I found personally SARS-CoV-2 and got sick for it. So the virus was probably from Rio de Janeiro, but I managed to get in Uberlandia. So coronavirus is as a final thing. There is, of course, not a single coronavirus, it's quite many, 54 if nothing has been added in the last couple of hours. So, and they are very diverse. Of course, once we are really working with viruses, pretty much everybody these days are trying to work with beta coronaviruses, and more specifically with some kind of SARS variant, SARS-CoV-2. So we didn't want to compete with a huge amount of people studying disease symptoms or studying the viral diversity or viral replicase proteins, because this is a quite tough work. We went a completely different way and started to look on RNA structures on a virus genome. This is just an example that RNA structures can be really, really important. This is not SARS-CoV-2. This is a classics. This is a classical iris element present in a genome of picornaviruses, and this is a 3' end of flaviviruses, which generates this kind of fancy RNA called flavivirus subgenomic RNA, which is very important for a virus. So if we draw RNA as a long linear molecule, it is a huge simplification. It really, in practice, is very, very highly structured. So my colleagues from Singapore took a kind of effort to find out how the structures in SARS-CoV-2 RNA look like. And using methods which I always pretend that I understand, but in reality, I don't understand them at all, they got a really nice mapping of the regions which have a very high base pairing. Of course, you can expect that a lot of these base pairs are just close range interactions, like stem loops or pseudoknots, and they are indeed very abundant. But to our quite big surprise, a lot of these interactions are spanning on a very large distances over the viral genomes, from 5' end to 3' end, from middle to the beginning, you name it. Really a big bunch of network. Of course, when you are seeing this picture, no, one thing is you are thinking, no. It's absolutely impossible that anybody designs rationally. It's beyond of that. It's truly complex. And then, of course, you ask, is it actually meaningful? Maybe it's just random. So we actually took effort and made a bunch of SARS-CoV-2 genetic mutants where we actually targeted several interactions, including this one, spanning to the distance, well, about 20,000 nucleotides over the viral genome. It looks like that. This is a duplex which holds it together. You can attenuate it or make it less stable by making a synonymous mutagenesis. No amino acid is changed, only nucleotides. And you can see that when you destroy this helix the viral replication goes considerably down. If you do another set of mutations, it's less so. If you do both, then the effect is quite mild because the duplex is restored. And you can do pretty much the same if you target the duplex with antisense oligonucleotide. Again, you will reduce the viral replication quite a lot. So this duplex exists in reality, and it is needed for virus replication. And at this point, I was really happy that we have found something. But our Singapore collaborations actually looked a bit detailed, and they analyzed their sequences. And they found out that the mutant virus we generated, this is this one, sorry for lousy quality of image, is unable to make mutations. It almost kind of doesn't have RNA editing, while the wild-type coronavirus mutates fast. This is a problem. This is why we have so big issues. Every year, many new variants. So what it is doing with this hairpin? Actually, it's very simple. To cut it short, this hairpin is a binding site of RNA editing enzyme, cellular enzyme ADAR1. It is bounding only when the stem loop is in logical, natural configuration. When we disturb it, the binding weakens. We can see that those variants in here bind it quite poorly. So this region is not only needed for replication, it's also a key element which allows the virus to mutate rapidly. Based on this data, you can conclude that alpha viruses, well, sorry, I'm not jumping to alphas, this will confuse only, that coronaviruses must have a very low tolerance for mutations which don't change amino acids, but do affect a nucleic acid structure. And indeed, we made 60 mutant viral genomes. This is another panel where we changed a number of codons to synonymous ones. And in most cases, we actually killed the virus. In some cases, virus survived, but in some of these cases where it actually survived, we observed quite significant attenuation, especially in this virus. You can see it makes much smaller plaques than a wild-type virus does. So what it gives us the idea, not unique, of course, to try it whether it works as a vaccine, which we obviously did. This is data from, I think it was from hamster model, but it's more or less the same in monkeys. If you give a wild-type virus, you see massive damage in lungs. But if you use attenuated virus, slightly attenuated virus, you can still see a damage. But if you use a strongly attenuated one, it looks not that different from uninfected tissue. So can it work as a vaccine? And the answer simply is yes. If you immunize hamsters with the attenuated virus and cage them together with the hamsters which carry a wild-type virus, the immunized hamsters never develop disease. We cannot see anything at all which look like symptoms, while the hamsters which were not immunized are naive. They develop severe lesions, which is typical for SARS-CoV-2 infection. So you can use the RNA structures, or actually removal of RNA structures, as an attenuation strategy of SARS-CoV-2 and presumably also many other viruses, not only coronaviruses, but also likely flaviviruses. Alpha viruses, it works really poorly. And here I stop. I just acknowledge few of funding agencies, and I'm happy to take your questions.
Thank you. Thank you very much. Well, that was a very, very interesting talk. So
unfortunately, I miscalculated.
No problem, no problem. So we have time for one or two questions. Let me allow to use the microphone here so people are trying to. I always forget how to do that. So here. Oh, here we are. So we have questions from Anna Carolini. How viral mutations can interfere with the formation of these secondary structures, and their stability?
Well, virus generates a mutation in a normal process. Actually, if you look on SARS-CoV-2 databases, you see that it has surprisingly few of those synonymous changes. Almost always the changes are changing also amino acid. So there is probably quite strong selection against the synonymous mutations, which I have not seen for alpha viruses, for example. How they affect it? I think that these structures have historically risen really a long time ago. And like in case of all RNA viruses are in constant kind of movement. It's most likely trial and error basis. Anything which is unfavorable for a virus is eliminated. Anything which is very favorable is maintained. Once it goes through the bottleneck, there is selection, random probably. So it's a normal process, it's the same which happens with the amino acid level. Just we should not forget that there is a several level of information. Information is present also in RNA structures, not only in proteins, what it encodes.
Perfect. We're gonna we have 3 questions in here, so I'm gonna answer by the order. So Carlos first.
Hi, do you listen to me, Professor?
Yeah.
Yes, hello. Thank you for the presentation. It's very interesting. So I want to know about the third C is about the RNAs, and about the microRNAs. Because, for example, in SARS-CoV-2, there are many microRNAs with great big impact on the infection. So in this case, in this another viruses, what do you could see about the microRNAs, or what we know actually? So is this? Thank you.
Yeah, this is a very complicated issue and very hot topic. Cellular microRNAs, the best known example obviously is hepatitis C virus, where microRNAs are needed for it to replicate. It is depending on the liver-specific microRNA. In most cases, the microRNAs have kind of supposedly a negative effect. It is not surprising that cell uses a microRNAs to conquer a virus, and we are really seeing that if we deliberately add a microRNA binding site to the viral genome, which is possible to do, then we can attenuate virus or restrict it from certain tissues. So there is also some data that viruses can actually encode microRNA-like structures. I mean, also RNA viruses, but I'm not very familiar to answer that. So currently, I would say that microRNAs are definitely important factors to restrict virus infection. And virus has active means to manipulate. Contrary to my old logics, I have actually seen, together with my colleagues, genomes of positive strand RNA viruses go to nucleus and do something there. And they do find specific microRNAs also in nuclear environment. So how they enter to the nucleus, what is all this for, I can't tell, but this is something which has happening.
Thank you very much, Professor.
Okay, thank you, Carlos. Professor Marcio.
Hi, Dr. Merritt, thank you for your great presentation. And I have been working with groups in US and Australia on development of vaccines using insect-specific virus.
Okay.
I would like to bring your points that you explained it very well to an integrative and One Health perspective. So then, my question is, in your opinion, can we target and conservative replication mechanisms across RNA viruses enable unified strategies for pandemic preparedness and antiviral drug development and vector-borne disease control? Do you think that is possible?
I'm not fully sure. I think that the insect-specific viruses have been overlooked for a long time. We have shown that the insect-specific viruses replicase is actually capable to engage the template of normal, so-called classical arbovirus, and work with it quite efficiently, which in my book means that they actually can recombine and can generate a very weird hybrids. Yes, the ones which have used as a vaccine, you probably are referring of flavivirus system and also the ELAT virus-based alphavirus vaccines. They don't produce virions in non-mammalian, non-insect cells, but they are nonetheless capable to produce replicase complexes in these cells. Alphavirus for sure. And I think this is a part of the reason why the vaccines are actually quite efficient, because they have a kind of small piece of the viral self ability to generate double-stranded RNA, for example, albeit in limited extent. So can it be any way evolved to the real virus? I don't know. Can we generate one drug which kills all alpha viruses? I guess yes. Polymerase is quite conserved, and we have many nukes which definitely are approaching that. Can we do anything which infects both alpha and flavi and other arboviruses? I would say no, because I have never seen a drug which can inhibit them all efficiently.
Okay, thank you. It's very, I could understand by now. So, and you think that we can just use for specific groups, like based on genus?
I think so. I don't think that it goes much beyond of that.
Okay.
Also, of course, there is always dreams that there is a magic bullet which will give them all down, but not going to happen.
Okay, thank you very much.
Thank you, Professor Marcio. Professor Rico. We cannot hear you. No, you have to activate your microphone. Not yet.
Okay. Each online program has its own peculiarities, and they tend to crash.
Yeah. So I think he's gonna type his question here. Yeah, we can do that. So yeah, it's not working. So there is another question, Orlando, without microphone. You need to turn on your microphone, Orlando. I think it's not working. I don't know why.
That's a computer.
Yeah, probably. So I'm gonna deactivate the microphones and activate again, so you can try it again. Okay, try it. Nope, it's not working. So I have one question, Dr. Merritt. So for the chikungunya virus, the one that you mutated the NSP4, so when you transfect the cell with the genome that is mutated, the cell expressing NSP4, so did you ever try to see how immunogenic is the virus that is produced by the cell? Because it's very interesting. It's not going to replicate anymore.
Yeah, it can. Actually, I haven't. But the Chinese group from Wuhan Institute of Virology in parallel made quite similar work. We published our papers almost the same time. And they indeed tried to use them as a vaccine, and the answer is that it is quite effective. So maybe more than the regular VLP vaccine is. Possibly owing very similar to that insect and arbovirus hybrid, that it can go to cell and do something. Also, it cannot make double-stranded RNA because of the lack of polymerase activity. So it is possible a vaccine, but it is more similar to VLP virus-like particle vaccine than a live attenuated vaccine.
Yeah, I wonder.
Okay, Orlando, we can hear you. So
Okay, now you listen to me?
Yeah.
yes.
Okay, great. Sorry about the previous issue. I have two questions. The first one is about your trans-replication system or mini genome. So is the expression of the reporter in the nucleus, so is it RNA pol-2 dependent, or is it done in the cytoplasm? Because I've seen the systems for this system for other viruses, and I was just curious whether this is expressed in the nucleus, so it's not capped, not polyadenylated, and it still makes to the cytoplasm for working as a genome.
Well, the RNA replication system seems to be exceptionally foolproof. We can launch it from RNA or DNA. It doesn't matter. It works in both cases. It requires the template to be polyadenulated, but it doesn't require it to be capped. You can make a lot of mix and matches games using different replicators from different viruses, different parts. In fact, we have generated currently a library. I think that it's 10 billion of different variants, combinations, and trying to screen it down to the most efficient one. So it's a kind of good tool to biotech, I think, in perspective. But thus far, we have mostly used it for basic studies.
Okay, and my second question, in regards of the formation of secondary structures that you showed in the end of the presentation, is ADAR1, the deaminase, targeting the genomes or transcripts? Because some of the secondary structures, they were very close by. So I was wondering whether this would be like hairpin for that is important for like transcription and translation, rather than something
absolutely, you are correct. Some of these elements are very well known what they are doing. They regulate replication. They are present in the 5' UTR. They are regulating this discontinuous transcription characteristic for coronaviruses. Almost certainly, they also regulate expression of several proteins from one and the same mRNA. So they are definitely multifunctional. But there are quite a few works just dedicated to study what their function are. They are just very numerous, and in most cases, the function is quite difficult to spot, especially those hairpins which are located, let's say, in middle of nowhere, like in the middle of viral open reading frames with no apparent influence on the initiation or termination or or ribosomal shifting or whatever similar mechanism. So yeah, they are definitely multifunctional, but I think that there are a subset of these RNA elements which are kind of more important, and most important almost certainly are these which are involved in RNA replication.
Okay, but do you see the deamination in the genome or both genome and transcripts?
In genome.
Oh, okay.
No, not transcript. If we are using, if you are meaning the transcripts made by viruses inside of the cell, those nested subgenomic RNAs, and then yes, the deamination is also present in there. But we specifically analyze the viral genomes for this editing score.
Okay, thank you.
Okay, thank you, everyone. So thank you, Professor Meritz, for this amazing talk. And I hope to see you again here in our Viral Club. So
It would be good.
okay,
Thank you for listening,
Okay, thank you so much. Thank you.
and all the best. You
Bye-bye.
have still a working day.
Yeah, bye bye.
Already night, starting to go very late in here.